Hello NEET aspirants. So yes, quickly let us recall all the diagrams of NCERT 12. So if we look into the major chapter, that is sexual reproduction of flowering plants beta, here we have to keenly focus on the very first diagram, that is male reproductive structure, that is stamens. Okay, male reproductive structure, those are stamens. Okay, stamens have a very long filament whose proximal end is connected to a thalamus, and distal end it has an anther which is dithecous. Okay, it has dithecous anther. Okay, dithecous anther. Okay, this dithecous anther, if we transversect that dithecous anther, you can find two lobes. Okay, two lobes of that dithecous anther, and each lobe has two theca, that is why it is called as dithecous. Okay, and it is bilobed anther. Most of the angiosperms will be having bilobed anther. The statement is important, Nana. The proximal end of the filament is attached to the thalamus and the distal end it is attached to a anther. Anther is bilobed and each lobe is having two theca, that is why we call it as dithecous anther. Most of the angiosperms are having dithecous anther only. Okay, and then if you see, each anther have the transverse section. If you look into the transverse section of each anther, you have 4 theca, right? What 4 theca, each is having a sporogenous tissue. Each is having a sporogenous tissue. So total 4 sporogenous tissues, and they have connective tissue at the middle. Okay, and this anther is having 4 layered wall layers. Okay, 4 anther walls. You can find 4 anther walls. Outer is epidermis. Next to that, endothecium will be there. Mostly endothecium is having alpha cellulosic layers or alpha cellulosic fibers. Next to that, you have 2 to 3 layers of middle layers. At last, we have inner layer, that is tapetum. Tapetum nucleus can be more than one, that is why we will say polyploid condition. Polyploid condition you can observe in the tapetum only. So all these 3 layers, the outer 3 layers will help only in one function. They constitutively perform only one function. What is that, Nana? First is protection of pollen grains, and second is, first is protecting the pollen grains, second is helping in dehiscence. Okay, so protecting the pollen grains and in dehiscence, these 3 layers will constitutively do that function. So outer is epidermis, next is endothecium filled with alpha cellulosic fibers, and then 2 to 3 middle layers, and then we have tapetum which is of polyploid condition. Okay, these 3 layers will constitutively perform one function, that is protection and help in dehiscence. So this sporogenous tissue will have microspore mother cells. Okay, sporogenous tissue will have microspore mother cells undergoing reductional division or meiosis, and they form 4 haploid microspores. They form 4 haploid microspores. Just a second. They form 4 haploid microspores. Okay, these microspores which are haploid will be matured to pollen grains. Okay, 4 haploid pollen grains. So this maturation, at the time of microspores maturing to pollen grains, the nutrients are supplemented from tapetum, and at sometimes middle layers also will be degenerated by giving nutrition to the pollen grains. Okay, so tapetum is keenly involved in providing nutrition to the microspores so that they will mature to pollen grains. By the time of maturation of pollen grains, 2 pollen sacs, they are formed into 2 pollen sacs, and by the line of dehiscence, this sacs will open and the pollen grains will be dispersed. Clear? So yes, this anther wall layers are very important beta, and sporogenous tissue. Ploid is at most important. Ploidy of all these layers is diploid. Okay, ploidy of entire anther is diploid, microspore mother cell is diploid. Undergoing reductional division, the ploidy of microspores and pollen grain is haploid. Do remember, Nana. Next, when it comes to pollen grain division, okay, inside that what happens is most of the pollen grains are released in two-celled condition. Most of the pollen grains are liberated in two-celled condition. Two-celled stage only pollen grains are liberated. So, this two-celled stage is formed by mitosis. This one pollen grain will undergo mitosis. That mitosis we will call it as amitosis, because it leads to the formation of asymmetric spindle, because it is due to the formation of asymmetric spindle. So, initially in the pollen grain, vacuoles, large sized vacuoles will be forming. Those large sized vacuoles will push nucleus to one peripheral end. So, this nucleus which is at the peripheral end, then the pollen grain will divide into two cells, forming an asymmetric spindle. This forms an what, Nana? Asymmetric spindle. After the formation of asymmetric spindle, two different celled stage or two different shaped cells are formed. Okay, one is large cell, that is vegetative cell, one large vegetative cell. This large vegetative cell has abnormal shaped nucleus. This large vegetative cell has abnormal shaped nucleus, and the small cell, that is generative cell, has spindle shaped nucleus. Okay, small cell that is having spindle shaped nucleus. Fine. So, yes, here you can see one large and one small cell. Large is having abnormal nucleus and small is having spindle shaped nucleus. And then if you see here, coming to the gynoecium, that is female reproductive structure. Female reproductive structure has stigma, long style and inside ovary. Okay, this is situated above the thalamus. If it is ovary is above the thalamus, that is superior ovary, and what type of flower, Nana? Hypogynous flower. If this is closed, syncarpous, all the ovary is fused, then it is called as syncarpous ovary. Best example is Michaelia. Best example is papaver. Okay, syncarpous ovary, best example is papaver. If carpels are free, okay, if carpels are free, then he is called as apocarpus. Okay, if carpels are free, it is apocarpus. Here carpels are fused, that is syncarpus. Example is papaver. Carpels are free, that is apocarpus. Example is Michaelia. Fine. And in most of the angiosperms, you can find inverted ovule, which is called as anatropous ovule. Okay, anatropous ovule, which is inverted ovule, which is found in most of the angiosperms. The funicle, which is called as stalk of the angiosperm, stalk of the ovule, funicle which is called as stalk of the ovule. This funicle and the spore, that is micropyle, they lie close to each other. Funicle and hilum, that is connecting the ovule to the stalk of the ovule, funicle. Okay, so the hilum and funicle lie close to each other. Fine. And here is the micropylar end. Opposite to that, you can find chaliceal end, and this is the outer integument, this is the inner integument, which is bitegmic, and inside the ovule you can find nucellus cells, and inside that you can see embryo sac. Embryo sac is haploid, female gametophyte of the angiosperm, and nucellus is diploid. Okay, all the layers or the parts of the ovule is diploid only. Nucellus is diploid, embryo sac is haploid. Next diagram, if you look into where megaspore mother cell at micropylar end, megaspore mother cell at micropylar end, at micropylar end. Megaspore mother cell at micropylar end will be differentiated or undergoing meiosis. It will form megaspore tetrad which is haploid. Okay, megaspore mother cell at micropylar end, it will undergo reductional division and it will form megaspore tetrad. That out of 4 megaspores which are linearly arranged, out of 4 megaspores, 1 megaspore which is towards the chalazial end is functional, the rest 3 megaspores will degenerate, they are non-functional. So, that 1 megaspore will undergo 3 sequential mitotic divisions. After first mitotic divisions, 2 cells are formed, one at micropylar end and one is at chalazial end. Second mitotic division, 4 are formed. Third mitotic division, 8 are formed, of which 1 nucleus from micropylar end, 1 nucleus from chalazial end will come at the center forming a central cell. Okay, they form a central cell. At micropylar end, you can find 2 synergids to mediate the pollen tube entry at this region and an egg cell above the synergid cells. Opposite to that, you have chalazial end where 3 antipodal cells are present, and at the center you can find 2 polar nuclei. And here if you see the embryo sac beta, embryo sac. In the towards the chalazial end you can find 3 antipodals, towards the micropylar end 2 synergids and 1 egg cell, and at the center you can see the central cell with polar nuclei. So, this is 7-celled 8 nucleated condition of embryo sac. Okay? I hope it is a very quick revision for you people. So, this diagram is about chasmogamous and cleistogamous flowers, where you can identify this in viola, where you can find this in viola, which is common pansy, and oxalis and commelina. Okay, viola, oxalis and commelina. In these plants you can find 2 types of flowers, those are chasmogamous flowers and cleistogamous flowers. Cleistogamous flowers are completely closed all the time, so they conduct only bud pollination or self pollination only. So, cleistogamous flowers conduct only autogamy. Whereas when it comes to chasmogamous flowers, this conducts both autogamy and geitonogamy. Okay? Geitonogamy is genetically similar or genetically self-pollination, but physiologically or functionally it is cross-pollination. Geitonogamy is genetically self-pollination, functionally it is cross-pollination. Clear ma? Understood clearly? Next. This is a plant like corn cob. This is what? Corn cob, which is mediating wind pollination. This is conducting wind pollination, wherein this elaborate feather like stigma or stamens which is open to the wind. Okay, they are exposed to the atmosphere, and through which the lightweight pollen grains which are non-sticky, they can easily fertilize or land on the stigma. And next, this is of Vallisneria or hydrilla. Okay, Vallisneria or hydrilla. So, these plants, okay, they conduct hydrophily, that is water pollination, wherein pollen grains which are released into the water, they are carried to the stigma by water currents. Okay. Here the pollen grain will be ribbon shaped. Okay, pollen grains will be ribbon shaped, and they can protect themselves from wetting of the water. Okay, this you have to remember. Vallisneria conducting hydrophily, and this is the diagram where the pollen grains are released into the water and they are carried to the female flower by water currents. And next, if you see abiotic agents pollinating like in Amorphophallus. Okay, just a second. amorphophallus, amorphophallus. This flower by fertilizing, okay, amorphophallus, that is fertilizing with yucca, okay, wherein here an insect, things like it is a, the flower will represent like a female bee, okay, female bee, and this will come and pollinate on that, okay. Pollen grains are dusted from the insect over the flower, fine. So this type of pollination is also seen in the abiotic agents, by biotic agents, where insect pollination is clearly seen, fine, where the insect will think like a female bee and it will come for the fertilization process, thereby it will dust the pollen grain also. And next, if you see, this is a very important diagram where we have to pay an extra attention, wherein the pollen grains landing on the stigma, a chemical dialogue will be there between pollen and stigma, wherein stigma surface should be receptive to the pollen grains by having certain sugar molecules, calcium ions and boric acid to mediate the journey of pollen tube all across the style, okay. So here the pollen grain, if one pollen grain is giving rise to one pollen tube, then we will call it as monosiphonous. If one pollen grain is giving rise to 2 or 3 pollen tubes, then we will call it as polysiphonous. So this will reach the synergids mostly through the this will reach the embryo sac mostly through the micropylar end, and it will release the male gametes. One will fertilize with the egg cell and the other will fertilize with the polar nuclei. That is why this is called as double fertilization and triple fusion okay. So here if you can clearly look into 2 male gametes where vegetative nucleus will be degenerated over here, and through filiform apparatus they are entering, they will first go to egg, one male gamete will go to egg and the other male gamete will fuse with polar nuclei, mediating the process of double fertilization. So here once the fertilization is done, the first product of fertilization, the cell or the product of fertilization is zygote. By continuous or sequential mitotic divisions, embryo is formed and the successive embryo stages are, first is globular embryo, next is heart shaped embryo, and then you can see with 2 cotyledons which are called as embryonic leaves. These 2 cotyledons which are called as embryonic leaves. Complete cotyledonary structure, dicot structure, you can see it over here. So what is the first embryo stage? Globular embryo followed by heart shaped embryo, then you can see a mature embryo with 2 cotyledons. Here if you can see beta, here you can see 2 cotyledons, right? Here you can see 2 cotyledons, just a second. Yeah, here you can see 2 cotyledons, and this is the plumule region, okay, this is the plumule region, from where you know this plumule will give rise to shoot, this plumule will give rise to shoot after development, after shoot development, and the terminal region over here is root cap and radicle will give rise to root, okay. If you can find 2 cotyledons, then it is a dicot embryo. This you can see here monocot embryo, where the papery cotyledon, that is scutellum, you can find it over here, and coleoptile, which is the covering sheath over the shoot apex or plumule, and here you can see coleorhiza above the radicle, fine, and this where a small fragment like structure above this is called as epiblast, okay. So all the labelings, keep it in your mind more keenly. And next if you see, first 2 cotyledons and this is the micropylar end, fine, and here you can see castor seed, fine, this is a castor seed. Castor seed is a dicot seed, only dicot seed, where you can see endosperm in that, okay, endosperm you can find only in the castor seed. See here you can see the endosperm, okay, from where you will get the castor oil, and this is of onion embryo, and this is monocot embryo. Many times questions asked from this. Recently in the EAP set 2025 also we got this question from the maze. Okay, so do remember we have a large embryo which is like with carbohydrate, filled with carbohydrate, and a proteinaceous embryo, starchy endosperm at the center and proteinaceous endosperm layer which is called as aileron layer. This is called as aileron layer. Below this you can see the papery cotyledon and a embryonal axis. Embryonal axis where above side it is plumule, covering the plumule this sheet is called as coleoptile, below it is a radicle, covering the radicle this sheet is called as coleorhiza. And next if you see Nana, apple, okay, false fruit, strawberry is also a false fruit. Why? Because this fruit is not just arised from a ovary wall, other floral parts are also involved, like apple, the main pulp of the apple is from thalamus of the flower, and here also you can see thalamus is involved, thalamus which is formed in the strawberry, also post fertilization you can see it in the fruit. Clearly understood, ma? So here what are the two false fruit, apple and strawberry, where thalamus is also involved in fruit formation along with the ovary wall. Okay. Now if you see principles of inheritance, show extra attention over here. This is the Mendel's traits. Okay, Mendel has taken 7 traits or characters. How many traits beta? 7 traits or characters. 7 traits or characters. 7 traits or characters. Okay, these 7 traits or characters, just a second. These 7 traits or characters have how many varieties beta? 14 varieties. They have 14 varieties. 7 traits or characters have 14 varieties. 14 varieties or variations you can say. So what are those 7 characters? Easily you can remember: seed shape, seed color, flower color, flower position, pod shape, pod color, and stem height. Okay. So if you see seed shape, the dominant is round variant and recessive is wrinkled. Seed color, it is yellow is dominant and green is wrinkled. Flower color, violet is dominant and white is recessive. Pod shape, full pod shape is like in flattened pod shape is dominant, constricted is recessive, and pod color, green pod color is dominant, yellow pod color is recessive. And then flower position, axial flower position is dominant, terminal flower position is recessive. And then stem height, if we take, tall is dominant and dwarf is recessive. As such we have 14 varieties. Okay, we have 14 varieties. All the dominant and recessive traits you have to fix it in your mind beta. They can ask your numerical question or any type of question based on any trait. Okay, it is not just like you will get a question on stem height only or only seed shape and seed color, nothing like that. You can get question from any of these traits. And then if you see here the emasculation process, first bagging, emasculation and rebagging will be done. Okay, here sometimes like in previous year question I have seen one question wherein how many times bagging will be bagging is done at the process of emasculation, one before the emasculation and other bagging after the emasculation. So just go through all the steps of the artificial hybridization also, and this comes from monohybrid cross. Monohybrid cross where pure lines are taken in the parental generation and after selfing you got a heterozygous dominant trait. Heterozygous dominant trait. Selfing the F1 generation gives you F2 generation, and in F2 generation from a monohybrid cross, phenotypic ratio will be 3:1, genotypic ratio will be 1:2:1. Clear Nana? Next. This is the Punnett square. Punnett square which is given by Reginald E. Punnett. Okay, Reginald C. Punnett, who gave us a Punnett square so that we can easily identify the genotype and phenotype of the cross, monohybrid or dihybrid cross. And here comes the concept of test cross. Here comes the concept of test cross and back cross. Test cross and back cross. Okay, back cross, if F1 generation is crossed with any of the parent, that is called as back cross. If F1 generation is crossed with only one of the parent, the F1 generation, that is dominant phenotype, here I will be writing here, it will be viewed by you, dominant phenotype crossed with recessive parent, dominant phenotype crossed with recessive parent. Okay, dominant phenotype crossed with recessive parent is called as test cross. Okay, this is called as test cross. Dominant phenotype, whether it is homozygous or heterozygous, is identified by test cross. So, mostly test cross is useful to identify the genotype of F1 generation. Keep that in your mind. If it is homozygous dominant, crossing on to recessive parent will give you all the violet flowers. If it is heterozygous dominant, then it will give you 1:1 ratio, where half of the flowers will be violet and half of the flowers will be white. That is how you will get 1:1 ratio. Fine. So, 1:1 ratio you will get if the F1 generation is heterozygous dominant. If the F1 generation is homozygous dominant, you will get all the violet flowers. And then if you look into Nana, incomplete dominance. Okay, incomplete dominance. Incomplete dominance. So, incomplete dominance is a concept wherein the dominant trait is not completely expressed. The dominant trait is not completely expressed, wherein red colored snapdragon flowers crossed with white colored flowers will give you pink color flowers which are heterozygous dominant. Okay? When they are selfed, you will get 1:2:1 ratio. You will get 1:2:1 ratio, where 1 is red, that is homozygous dominant, 2 are pink, that is heterozygous dominant, and 1 is red, that is homozygous recessive. So, here in the incomplete dominance, you can see same phenotypic and genotypic ratio, and this is an exception of Mendelian principles. Okay. Now, now comes the concept of multiple allelism, that is ABO blood grouping. ABO blood grouping, here we have 3 alleles. Okay. ABO blood grouping has how many alleles beta? 3 alleles. ABO blood grouping, we have 3 alleles, that is IA, IB and IO. Okay, these 3 alleles are there. So, IA, when it is crossed with parent to IA allele, then you will get A blood group. IA with IB then is AB blood group. IA and IO gene or I gene, that also gives rise to A blood group. So, here AB blood group, both the alleles are dominant, So, here comes the concept of codominance, incomplete dominance, codominance and multiple allelism. All these 3 are what, Nana? Expressions. All these 3, like multiple allelism, codominance or incomplete dominance, all these 3 are the exceptions of Mendelian's laws. What law is that? They are the exception of Mendelian law of dominance. They are the exceptions of law of dominance. Okay. Whereas, law of segregation that is given by Mendel is universally accepted. Fine. Now, the same applies to IB blood group, and IO gene will give you O blood group. As such, you will get 4 phenotypes and 6 genotypes in ABO blood grouping. Very good afternoon, Rajnikant. Then it comes to dihybrid cross. When it comes to dihybrid cross. So, here in the dihybrid cross, if you see, the dominant parents, homozygous dominant and homozygous recessive, where two traits are taken, okay, that is seed color and seed shape. After F2 generation, of course, the selfing of F1 generation, that is heterozygous dominant, crossed with capital R, capital Y, small y. On self fertilization, we will get the phenotypic ratio 9:3:3:1. This is the phenotypic ratio. So, I will give you a small shortcut where you can easily solve the numericals over here beta. Concentrate more here, wherein out of 9 dominant, okay, out of 9 dominant, one is homozygous dominant and one is homozygous recessive, fine, and rest 4 are heterozygous dominant, rest 4 are heterozygous dominant here, and 2 are with one heterozygous and one homozygous condition. Rest 2 again with one homozygous and other heterozygous condition. Clear? And 3 if you take one homozygous like, and rest 2 with heterozygous condition. Okay. Next 3 if we take again, one is homozygous dominant and homozygous recessive, you can see, and one again rest 2 heterozygous dominant and homozygous recessive. If there is any question regarding the dihybrid cross, and you can keep this in your mind, okay, if you can keep this strategy in your mind, you can solve it easily. Like here you can get easy numbering of for 16 zygotes, okay. How you will get the number of zygotes? The number of zygotes we will get by gamete square, okay. Number of zygotes we will get by gamete square. Here how many gametes do we have? How to calculate the number of gametes in the F1 generation? If you take capital R, capital Y, small y, types of gametes we can identify by the formula 2 to the power of n, where n is the number of heterozygotes, right. So, here how many heterozygotes are there beta? 1 and 2, 2 heterozygotes. 2 to the power of 2 is 4, okay. So, 4 types of gametes are there. 4 square will give you number of zygotes, that is 16 zygotes. For 16 zygotes, these are the types of genotypes that you are getting. For 520 seats, for 210 seats, for 200 seats, what is the number of such genotype they will be asking. If this is in your mind, you can solve that type of questions very easily. So, as of now, do remember Nana, tomorrow in your exam, if there is any question based on types of gametes, then apply the formula 2 to the power of n. If there is any question based on number of zygotes, then apply the formula gamete square. If there is any numerical question based on the dihybrid cross, then just look into this and just apply this shortcut, you will get into it. And next, if you see, this is the prior experiment, or at the time of cell cycle discovery, that is prior to chromosomal theory of inheritance, like how the chromosome pattern is moving all across the cell cycle. G1 phase, in S phase DNA duplication happens, that you can see easily, where DNA is doubled but number of chromosomes are same. First in meiosis 1 in anaphase, these 2 chromosomes without any centromere splitting, the homologous chromosomes, 2 chromosomes will move apart, okay. In anaphase 2 of meiosis, centromere splitting takes place, and rest at the end you can find 4 different types of daughter cells. Clear? So, this is how uniting the cell theory with Mendelian's principles, they gave us chromosomal theory of inheritance. And here if you see the concept of linkage and recombination, the concept of linkage and recombination, which is given by Morgan, okay, Thomas Hunt Morgan. So, this linkage and recombination, if you see Nana, in the parental generation, he has taken 3 traits, okay, 3 traits if you see of the female parent and male parent, okay, male parent. Of course, Y chromosome will be shorter than X chromosome, but here I will take similar so that you can mark it easily, mark the traits easily. So, one trait is with respect to, it is with respect to body color, and second is with respect to eye color, and third is wing shape. Third is what, Nana? Wing shape. Okay. So, here the naming or the alphabet is given with respect to the recessive traits. The recessive trait for the body color is yellow. So, it is Y, and the dominant is brown. So, he gave Y plus. Okay. Recessive trait for eye color is W, and the dominant trait is W plus. Okay. And wing shape, if you see, the recessive trait for wing shape is miniature wing, dominant is M plus. Clear? These are the traits, and if you can clearly observe beta, the distance between these two genes where body color and eye color are closely linked to each other, when two genes on the same chromosome are linked, then there is no possibility of crossing over. There is no possibility of crossing over. So, yes, here if you see, when these two are crossed, body color and eye color, if you see body color and eye color in cross A, when they are crossed, you can observe more amount of parental generation because the distance is small, crossing over is very less. Therefore, recombinant types are only 1.3%. If you look into cross B, the cross is in between eye color and wing shape. The cross is in between eye color and wing shape. As the distance between the genes on the chromosome is far, then you can easily crossover happens. As the distance is far, crossover can happen, as distance is directly proportional to crossing over or recombination. Therefore, here the recombinant types are 37.2 and parental generation is just 62.8. Keep that in your mind, fix it in your mind. Fine. This is the pattern. If you can draw this and you can analyze easily, that will be fine beta. Coming to pedigree analysis. If you can identify the diseased trait in each and every generation, then that is dominant. Okay. And here, as it is applied, as there is no crisscross pattern of inheritance, then we can go for autosomal dominant disease. Autosomal dominant disease. Once after finding based on the basic features, okay, we have identified this pedigree with basic feature, that is what? Autosomal dominant feature, right? That is, the disease is observed in each and every generation, and then we cannot find any crisscross pattern of inheritance. So, we are going to take autosomal dominant only. So, in this autosomal dominant trait, let us apply and see. This is expressed both in the homozygous and heterozygous condition, right beta. So, here I am taking it as such. So, what are the possibilities of that? Capital A, small a, capital A, small s. This will be the possibilities. So, here again it is, let me take it heterozygous only, and this is diseaseless, that is homozygous. So, here again you can, there is a possibility of getting one deceased offspring. So, here the disease that is autosomal dominant is myotonic dystrophy, and here there is no disease in the parental generation, and you can see it in the first generation, that is F1 generation, and here also there is no crisscross pattern of inheritance. There is no disease in the parents, but you can see in the offsprings, and here you can see the skip of generation also. So, this is autosomal recessive, that is sickle cell anemia. What is it, Nana? Sickle cell anemia. Sickle cell anemia. Clear? Understood? And then if you proceed, yeah, sickle cell anemia is an autosomal recessive mutation, where you will also call it as point mutation, that is because of the change in the single base, single nitrogenous base, change in just single one nitrogenous base, that is at the place of GAG, at the place of A, here you can find T, okay. That is how it is given, like again in the place of GAG, here it is GUG in the mRNA, for the translation, in the place of GAG, which is coding for glutamic acid, that is 6th amino acid of alpha globulin chain, HbA protein, okay. That is which amino acid beta? sixth amino acid, that is coding, GAG is coding for glutamic acid, that is just by change in one base, that is adenine to uracil, that is GUG, it is coding for valine, which gives you sickle shaped red blood cells, sickle shaped red blood cells, which creates oxygen tension. As oxygen transport is not done in a proper way, oxygen tension is created. That is how the oxygen transport is affected because of the change in the shape of red blood cell, that is sickle cell anemia. And then if you see, that is chromosomal disorder, Down syndrome, okay, Down syndrome chromosomal disorder, that is because of trisomy of 21st chromosome, trisomy of 21st chromosomes. The symptoms you can see is broad flattened face, big wrinkled tongue, congenital heart, palm creases, you can see palm creases, and flat back head, you can see mental retardation, frequent micturition. All these are the symptoms of the Down syndrome, that is because of trisomy of 21st chromosome. Let me discuss two more diseases also, that is Klinefelter syndrome and Turner syndrome. Klinefelter syndrome is observed in males, that is because of trisomy of allosomal chromosomes, that is you can see 2 X chromosomes, that is 44 plus XXY. And if you see Turner syndrome, that is XO1 X chromosome will not be there in Turner syndrome, and Turner syndrome is seen in females. In Klinefelter syndrome you can observe gynecomastia, in Turner syndrome you can observe rudimentary ovaries, where both the individuals of Klinefelter syndrome in males and Turner syndrome in females are sterile. Okay. And then if you see Nana, molecular basis of inheritance, carefully you observe, this is the single stranded DNA from 5 prime to 3 prime. First we will understand the nucleotide. Nucleotide has a sugar molecule at C1, C2, C3, C1, C2, C3, C4 and C5. Pentose sugar, as this is the DNA strand, this is ribose, okay, the sugar is deoxyribose, as it is DNA strand, the sugar is deoxyribose. If it is RNA, what is the sugar, Nana? If it is RNA, that is ribose sugar. So at 3 carbon, at third carbon, OH group is present, okay, OH group is present, which is connecting the successive nucleotide. So for a single nucleotide formation, first nitrogen base attaching to first carbon, if the nitrogen base is purine, the N9 of purine will attach to C1 of the sugar. If the nitrogen base is pyrimidine, the N1, nitrogen 1 of the pyrimidine will come and attach to carbon 1 of sugar. So this linkage is called as glycosidic bond. This is glycosidic bond, okay. And the link between the sugar, this nucleoside, the nitrogen base and sugar will form nucleoside, nitrogen base and sugar will form nucleoside. Nucleoside plus phosphate group at 5 prime end or 5th carbon of the sugar is by phosphoester bond. This is by phosphoester bond. So here what you have to remember, beta, for the formation of one nucleotide, we need one glycosidic bond and one phosphodiester bond, and for the formation of nucleoside only glycosidic bond is enough. For the formation of 2 nucleotides we need phosphodiester bond as well. Phosphodiester bond is in between 3' carbon of one nucleotide and phosphate group, which is at the 5th carbon of the successive nucleotide. So for a dinucleotide, how many bonds we require, beta? Glycosidic bond, phosphoester bond and phosphodiester bond. So for a single stranded DNA, all the linkages that are required are, glycosidic bond, phosphoester bond and glycosidic bond, phosphoester bond and phosphodiester bond. These are the 3 bonds that are required for the single stranded DNA formation. For the double stranded DNA, one extra bond is required, that is hydrogen bond. Hydrogen bond. If it is in between adenine and thymine, 2 hydrogen bonds are required. If it is in between guanine and cytosine, 3 hydrogen bonds are required. And then base pairs, okay. Here this is the double helical structure of the DNA. The distance between two base pairs, is 0.34 nanometers or 3.4 angstroms. This one turn, from here to here it is one turn. This one turn is called as one pitch, okay. One pitch or one turn of DNA will have 10 base pairs, and the length is 34 angstroms. The length is 34 angstroms, and one strand of the DNA is from 5 prime to 3 prime, other strand of the DNA is from 3 prime to 5 prime. They are anti-parallel to each other or complementary to each other, okay. Do remember beta, most important. Now when it comes to molecular basis of inheritance, that is central dogma of molecular biology is very much important, Nana. Definitely you will get a question from this tomorrow, okay. For sure I am saying central dogma of molecular biology. For 2 minutes listen carefully. First is replication, okay. First is DNA replication. First we will consider prokaryotes and then we will look into eukaryotes. For replication we need DNA dependent DNA polymerase enzyme, and once the DNA is replicated in the transcription process, for this we need RNA polymerase in prokaryotes, and then mRNA is synthesizing proteins. This is translation. All the steps takes place in the cytoplasm itself if it is in a prokaryote. If it is a eukaryote, DNA replication takes place inside the nucleus, and DNA dependent RNA polymerase 2 will give rise to mRNA. Apart from this RNA polymerase 2, we need 2 more polymerases. Those are RNA polymerase 1 and RNA polymerase 3 as well. RNA polymerase 1 will synthesize rRNA, RNA polymerase 3 will synthesize tRNA. Keep that in your mind, okay. So here RNA polymerase 2 will synthesize mRNA, and the ultimate goal of DNA is to synthesize the proteins. mRNA will be translated to proteins. So both up to replication, transcription, post-transcriptional events takes place inside the nucleus. Only translational events takes place in the cytoplasm if it is a eukaryotic cell. In few viruses, like for example retroviruses, if RNA is the genetic material, then first this RNA have to be converted to DNA, that is by the enzyme reverse transcriptase. After this, then the process will be operated, okay. So this is central dogma of molecular biology. Do remember all the labelings. In one of the PYQ question, directly from this the question is appeared, okay. And next, if you see from packaging, there is high possibility of question from this area also beta. So here in DNA packaging, DNA is negatively charged. It is wrapped around positively charged histone octamer, wherein histones have H2A, H2B, H3 and H4. This is 2 such pairs, okay. 2 pairs of this histone proteins will form a histone octamer. This will form a histone octamer, okay. So histone proteins are positively charged. Around this histone proteins, DNA will be wrapped around this histone proteins. DNA is wrapped around this histone proteins. That is how condensation of DNA will be taking place. And the histone proteins are rich in the amino acids. Those are lysine and arginine. Clear beta? Done. And next, if you see, this is Hershey and Chase experiment where they gave unequivocal proof that DNA is the genetic material. Remember, here they have taken 2 bacteriophages, one with an isotope of sulfur at the protein coat and one with a labeling of phosphate P32 in the genetic material. After infection into a bacteria, after blending and after centrifugation, they have observed that the supernatant has S35 isotope, whereas in this, wherein P32 is there, the supernatant, they do not find any radioactive phosphate. It means the radioactive phosphate has entered the genetic material of bacteria. This has entered the genetic material of bacteria. That is how they proved that DNA is the genetic material that is passing. Next, this is the experimental proof of Michelson and Stahl. Many a times, 2 to 3 times the question is asked from this beta. So, this Michelson and Stahl's experiment is done on the model organism, that is E. coli, to prove the semi-conservative method of DNA, wherein E. coli one generation is 20 minutes. In E. coli, one generation is 20 minutes. Okay. As such, in each generation, both the strands of the DNA will act as template strands and they will synthesize 2 daughter strands. Both the strands will act as template and they will synthesize 2 daughter strands. As such, if you can see, after 60 minutes, you can see 2 hybrids and 6 light chains. Okay, 2 hybrids and 2 light chains. Fine. This is how you can observe that. Many calculation questions are done in our previous sessions also. Once go through it. And then this is the semi-conservative or semi-discontinuous DNA replication, wherein one is from 3 prime to 5 prime, another is from 5 prime to 3 prime, wherein both the strands are acting like parental strands. Leading strand is towards the replication fork. Always keep in your mind beta, synthesis of DNA or RNA is from 5 prime to 3 prime only. So, from 5 prime to 3 prime, leading strand is towards the replication fork, lagging strand is away from the replication fork. So, lagging strand will form like Okazaki fragments. Okay, the small RNA primers, those are what? Okazaki fragments. They are formed in a discontinuous fashion. These are Okazaki fragments. Fine. At the last step of DNA replication, they are binded by DNA ligase. So, first this is the double stranded DNA is opened by DNA helicase, which is ATP dependent process, and then RNA primers are joined at the 3 prime end, of the parental strand. RNA primers are kept by the enzyme primase, then DNA polymerase will remove these primers, they will remove this RNA primers and they will start synthesizing DNA strand. Okay. Leading strand is towards the replication fork, lagging strand is away from the replication fork. Finally, this Okazaki fragments are adjoined by the DNA ligase. DNA ligase is also an enzyme is ATP dependent process. DNA ligase is also ATP dependent process. And here if you see, you have to note the template and coding strand. Template strand and coding strand, if you see, this is the parental strand and this is the daughter strand. For example, okay, at the coding strand, the 5 prime of the coding strand will be the region of promoter, and at the 3 prime region of the coding strand will be the region of terminator. In between you can see structural gene. Okay, 5 prime end is promoter and 3 prime end is terminator, and at the center you can see structural gene. Fine. So, here one promoter, structural gene and terminator will form one transcriptional unit. This will form one transcriptional unit. This forms one transcriptional unit, promoter, structural gene and terminator, and mRNA strand whichever is synthesizing, that is from 5 prime to 3 prime direction. It is complementary to the template strand and same as that of coding strand. It is same as that of coding strand. So many times the question is asked. So, look into the sequence and then code. If the mRNA strand is given, you have to choose if the template strand is given and you are asked to find out the mRNA, copy for a complementary strand. If coding strand is given, then it will be mRNA strand will be same as that of coding strand, except T in the place of coding strand, here you will have uracil, that is it, rest everything is same. Here if you see the transcription process, first RNA polymerase along with sigma factor forms a holoenzyme that will open the double stranded DNA, forming a transcription bubble. This will form a transcriptional bubble, okay. And here if you see, after elongation, the sigma factor and RNA are released, the sigma factor is released out and RNA polymerase is binded with rho factor and this is pulled out. So, at the termination RNA polymerase is binding with rho factor, at the initiation RNA polymerase is binded with sigma factor, keep that in your mind. And if it is a eukaryotes, in eukaryotes you will find post transcriptional events. Post transcriptional events also takes place in the nucleus, wherein at the 5 prime capping takes place, capping of methyl guanosine. At 3 prime you can see polyadenylated tail and splicing at the center where introns or inactive sequences are spliced, okay. 3 post transcriptional events, one is 5 prime capping of methyl guanosine, third at 3 prime and you can find polyadenylated tail, and in between you can see splicing which is mediated by SNRPs, okay, where that is RNA and proteins, few RNA and proteins which are SNRPs, they will cleave the inactive sequences, those are introns. So, which mRNA will be travelled from nucleus to cytoplasm for translation ma? The one which has 5 prime cap and only exon sequences, at 3 prime polyadenylation, that sequence is travelled or given to the cytoplasm. And then if you see genetic code, it is very simple. If there is any question given for you from genetic code, just take 2 minutes of time, AUG you already know, AUG is a start codon that codes for methionine, that you know, and 3 stop codons UAA, UAG and UGA, these 3 are stop codons, this also you know. Now if you see the successive codons, other codons how you have to remember is the first codon, 3 triplet will form 1 codon, right. So, the first base is from this end UCAG, remember the order, second is from this line UCAG and third is from this line of each box UCAG. As such fill 4 boxes, and then this is first 2 are phenylalanine, next 6 leucine and the next 3 isoleucine, last box is of valine. Second column is easy to remember, serine, proline, threonine and alanine. Third if you see, those are 2 amino acids, tyrosine, histidine, glutamine, aspargine, lysine, last is of acids, aspartic acid, glutamic acid. Fourth is also easy, that is cysteine, tryptophan, then 6 arginine interrupted by 2 serine, last box is of glycine. Like that you can remember it easily, it is just a 2 minutes process. So, do not miss a question if it is coming from genetic codon. Last is translation beta, and sometimes the questions are asked like what is the first step of translation. The very first step of translation is amino acylation of tRNA and charging of tRNA. Amino acylation of tRNA, when the tRNA is charged, at 3 prime, and when amino acid is added over here, when it is charged, when the tRNA is charged, that is amino acylation of tRNA. This tRNA will is the first step, charging of tRNA is the first step for translation, then it will come and sit on the mRNA, over the mRNA, first smaller subunit of the ribosome will come, later larger subunit which has 3 sites, those are A, P and E site, okay. tRNA landing on the P site, it will synthesize amino acid, and this P site will synthesize a protein, that is peptidyl transferase, which is helpful in the formation of peptide bond between two amino acids. As such, a polypeptide chain is formed. Clear? So, this is how a polypeptide chain is formed. Each tRNA will code for one codon. So, all the 64 codons eliminate 3 stop codons, that is 61 codons have unique tRNAs, all different types of tRNAs. One tRNA is unique for one codon. Each will come sit and that will give rise to amino acid and escape from the exit site. Next, this ribosome movement across the mRNA is called as translocation process. Ribosome will move all across the mRNA and tRNA will code for amino acids. This is how it happens in the translation process. Okay. Now, finally, the last slide, which is the most important slide, where gunshot question comes from this concept, that is gene expression. Not even a single paper is escaped from this concept, beta. So much important, that is gene expression. In gene expression, if you can observe from 5 prime to 3 prime, 5 prime to 3 prime, that is in the mRNA, okay, 5 prime to 3 prime in the mRNA strand, you can observe promoter region at the 5 prime end, right. So, to promoter region, do remember always RNA polymerase will come and bind. What will bind, beta? RNA polymerase will come and bind to the promoter region, which will initiate the process of gene expression. Then this I gene will synthesize repressor mRNA. I gene will synthesize repressor mRNA. Okay, it will synthesize repressor mRNA. Next, promoter region, to this again RNA polymerase will come and bind. Repressor, if it is free without any lactose, then it will go and bind to the operator region, inhibiting the process of gene expression. Here the inducer is lactose. If the culture of bacteria is given with lactose, then this lactose having the promoter region, RNA polymerase will bind, RNA polymerase will bind to the promoter region, then I gene will synthesize the repressor, excuse me, I gene will synthesize the repressor, then again to the promoter region, RNA polymerase will bind. To the operator region, their repressor will not bind because lactose will bind to the repressor and repressor will be inactivated. So, nothing is there here to block the process of gene expression. So, here ZYA, 3 structural genes are present, where Z is coding for beta galactosidase, Y for permease and A for transacetylase. Beta galactosidase excuse me, it will cleave the lactose to glucose and galactose. Lastly, Y gene, which is coding for permease, this will increase the permeability of lactose into the cell. Transacetylase will change the acetyl group and it will increase the solubility of absorbing the sugars into the cell. So, this is all about the gene expression, beta. Remember only one gunshot thing, that is to the promoter region, RNA polymerase will bind, I gene will synthesize the repressor that binds to the operator region. Repressor will be inactivated by the inducer, inducer here is lactose, and it has 3 structural genes, that is ZYA, Z codes for beta galactosidase, Y for permease and A for transacetylase. What is the function of beta galactosidase? Cleaving the lactose into glucose and galactose. Permease will increase the permeability of lactose into the cell. Transacetylase will change the group of, it will give the acetyl group where it increasing the solubility of the sugar in the cell. This is all about the gene expression. So, with this we have covered the major concepts, beta, concepts along with diagrams, which is highly useful for your tomorrow's exam. Just have a glance of this and go, you will never regret watching this class. Okay? All the best for your exam.